Okay, I just wanted to post this PowerPoint lecture with the slides intact and all that because I realized on Wednesday I had to rush through some of the later parts of the lecture, and I just wanted to go through in a more appropriate time scale and talk about some of these things. Because we spent an awful lot of time talking about the architecture, the structure of a action potential and how that works in terms of voltage-gated sodium channels and voltage-gated potassium channels. So I just want to, it's about a dozen more slides here, and just go through them with you. So again, an action potential is an all or none event. Once that critical threshold potential is reached, it's about 10 millivolts more positive than the resting membrane potential. Once that is reached, you have an action potential. And the action potential will be the same in terms of its amplitude, the size of that depolarization, and the time. The action potential is self-regenerating, which is critical then, because once the action potential is initiated, and give me a moment here, let me see if I can make a pointer here. Okay, hang on, I'm going to actually leave. How does that work? Slideshow. Let's try this again. We'll go with the pen. Okay, so once the action potential is initiated, right here at the axon hillock, then basically it will travel unidirectionally, and that's again because of the refractory period of the channels after they've opened and closed. And so what you can see then is that this is self-regenerating. Once it's initiated at the axon hillock, it will travel the length of the axon until it gets to the very end. And again, size of the stimulus will not affect the size of the action potential. It will always be, some constant amount of depolarization, typically something from -70 resting membrane potential up to +30. The size of the stimulus will not affect the duration of the action potential as well. So again, just to stress this, the coding of intensity in terms of stimulus is going to be coded by frequency. Okay, so here we have basically, I have to keep, sorry. Keep doing this, huh? Here we have no action potential generated, and here we have a stimulus that will give us some level of awareness of whatever the sensation is, if this were a sensory physiology mechanism. And then over here we have a higher, more intense signal, and it's coded again by the frequency. So the reason why they're unidirectional, again, has to do with this refractory period. So there's two refractory periods. There's the absolute and the relative. The absolute really is just to say you're not going to have an action potential while you're already having an action potential. The relative refractory period has to do with the fact that there is hyperpolarization going on, and so you can see in this period over here of the action potential, what you have then is membrane potential actually going to be more negative than it was at resting. And so what you have then are potassium channels still open, which means really there is not the reset necessary to initiate a new action potential unless the stimulus is very, very strong. And again, we went over this, but this is just sort of what the confirmation of those all-important ion channels look like during different points of the action channel. So we have our leaky sort of always open potassium channel, we have our voltage gated potassium channel, and then we have our voltage gated sodium channel. So during the depolarization stage, the time when we're going from -70 to much more positive membrane potentials, the sodium channels are opening, responding to the change in the voltage, the membrane potential, and it's sort of a positive feedback loop. As some of them open, well then it gets more depolarized and more of them open, and then it gets more depolarized and more of them open, and that continues and continues and continues all the way up to about +30 millivolts, because at some point you reach that sensitivity portion of the voltage-gated potassium channels. And so when that happens, you have potassium leaving, and the reason for that again is because of the electrochemical gradient. So now we're at a very different membrane potential, and so what happens here is that enormous chemical gradient, the diffusive gradient, the 140 millimolar potassium inside the cell relative to about 5 or 4 millimolar outside, well, now potassium flows out of those channels. We're losing positive charge, so we're reestablishing that negative membrane potential. And again, this is the refractory period. This is when the sodium channels are shut down, and so they will not open up again, and therefore you are forcing the action potential to travel in one direction. Myelination, extremely important for most of the nervous system, both the peripheral and the central. So basically we have these cells that will wrap themselves around the axons, and they act like insulating wire. And the little gaps in between them are the nodes of Ranvier, and this is where the axon is not covered by myelin, and this is basically where action potentials will be generated. And this is where all of that molecular machinery, the sodium channels, the potassium channels, the sodium-potassium ATPase are invested in those areas, and that's where action potentials occur. And then the action potential will jump to the next node of Ranvier in that unidirectional way it's traveling, and we call that saltatory conduction. Okay, and that's just shown here. And again, what we have then in the peripheral nervous system are Schwann cells that are providing the myelination, and then in the central nervous system we have the oligodendrocytes that can do that. And the important thing you can see here is one Schwann cell will wrap itself entirely around one portion of an axon, whereas an oligodendrocyte can reach out these dramatic cytoplasmic extensions and wrap themselves around several axons simultaneously. Okay. And again, the importance of myelination and saltatory conduction can really be understood when we look at pathologies related to the malfunction of that. And one dramatic example of that would be multiple sclerosis in the central nervous system when myelination is lost. And just to give you some numbers, the importance of having the saltatory conduction and having all the machinery invested at high density at these nodes of Ranvier, you can see the insulating properties. So an unmyelinated fiber can only conduct at about 1 meter per second. We see this in slow visceral responses of the smooth muscle system, and myelinated fibers are 100 times faster. So you can really see just how important this myelination is. Okay, so neurons, when they communicate with other neurons, they form a synapse. We have two basic kinds of synapses. The chemical synapse, which is probably the synapse you guys are familiar with if you have any familiarity with this, and then the electrical synapse. The electrical synapse is really the exception, and the only importance in terms of mammalian physiology is for cardiac cells. And this is the way the muscular tissue that makes up the cardium, the myocardium, the base of the muscular tissue of the heart, they are electrically connected. They don't have chemical synapses, they have electrical synapses. And so what you have then is basically a action potential that will travel through the cell, and then it will physically move through these gaps, these connections that connect one cell to another, and the action potential will generate itself from one cell to the next cell that way through this very tight physical connection. And again, we only worry about that in cardiac cells. But this is a great way to make sure that the cardiac cell is working, or the heart is working in unison, and all the cells are conducting at the appropriate time. And so we call these gap junctions. And so again, the pre- and postsynaptic membranes are only a few nanometers apart. These very important molecular pores called connections form this connection point between the cytoplasm of two cells and allow ion flow. Transmission is extremely fast, but it's also bidirectional. And electrical synapses are less common, again, because they don't give you a lot of the regulatory power you'll see in a chemical synapse. There isn't temporal summation. You need a lot of contact area, and they are always excitatory. They cannot be inhibitory. So chemical synapses, one of the major ones we see, are at the neuromuscular junction. This is basically between a motor neuron and the skeletal muscle that's being controlled by that motor neuron. And the motor neuron will release the neurotransmitter acetylcholine from its axon terminals, and the postsynaptic membrane of the muscle, this motor end plate, will basically respond to the acetylcholine in an excitatory sense. And this will cause muscle contraction. So the synaptic cleft is a space between the pre- and postsynaptic membranes. The action potential causes the neurotransmitter acetylcholine to be released basically by opening up voltage-gated calcium channels, and then calcium enters the axon terminal, and that causes then vesicles holding acetylcholine to be released from the presynaptic neuron into the synaptic cleft. Okay, and that's shown here. And so again, what we have, let me get the pen going here. What we have is the action potential traveling down the cell. And again, that's being propagated because sodium channels are opening, causing depolarization. That depolarization is traveling down the cell. And when it gets to the very end, at the terminal bouton, basically it's now going to activate sodium channels, as you see here, but it's also going to activate calcium channels. And again, the electrochemical gradient of calcium is much like sodium in that when a calcium channel opens, calcium will come flooding into the cell. The flooding into the cell of calcium will then basically cause these vesicles holding the neurotransmitter, in this case acetylcholine, to make their way to the membrane of the presynaptic neuron and be released. And so the acetylcholine will then be released into the synaptic cleft, an extremely small distance, about 20 nanometers, and now it's all up to the diffusive forces for the acetylcholine to diffuse across this space and then bind to a receptor on an acetylcholine receptor molecule. That receptor will basically open up a sodium channel, and sodium will rush into the postsynaptic cell, in this case a muscle cell, causing depolarization, causing an excitatory response. So basically a excitatory postsynaptic response, an EPSP. So the neuromuscular junction, in this case, it's always using acetylcholine, and it's always causing depolarization because it's opening up a sodium channel. Sodium channel is basically, always remember that, sodium is going to equal depolarization. Look at this handwriting, it's fantastic. Anyway, we'll just leave it at depolarization, depol. Okay, because sodium is going to rush into the cell and it's going to cause the resting membrane potential to become less negative. That is the definition of depolarization, and that is always excitatory. So in this case, excitatory means we get the muscle cell to flinch. And we'll talk more about muscle physiology in a few chapters. Also, just to, again, stress the importance of synapses where neurons talk to other neurons. We have 500 trillion synapses in the brain. And again, that means 1 billion synapses per cubic millimeter. That's an incredibly large number, very hard to even appreciate what that means. So, how do the neurons communicate with other cells? Again, postsynaptic membranes respond to acetylcholine, and that is the one sense where once we have the action potential and it causes the release of the neurotransmitter, we are now left to the simple process of diffusion. There's no way to actively move the neurotransmitter across the synaptic cleft, which is why the cellular communication, the cellular architecture between a pre- and postsynaptic neuron, they're extremely close to each other. So the diffusive gradient will almost instantly carry the acetylcholine from the presynaptic neuron across the synaptic cleft to the postsynaptic neuron. Okay, and this allows basically sodium and potassium to flow through and increase. And in this case, what we have here is predominantly sodium. Okay, whoops, let me go back. Sodium will flow through this channel, and so the acetylcholine receptor is also the ion channel. This will be important in a second. Okay, and so the ion channel opens. In large part, sodium moves through depolarization. Now, we can have graded potentials. Okay, so when ligand-gated ion channels open, the membrane potential changes depending on which ion channel is open. So if you open a sodium channel or a calcium channel, you cause depolarization, and that is excitatory. So we call that an excitatory postsynaptic potential, an EPSP. Now you can also have neurotransmitters that will travel across, and they'll cause the opening of a potassium channel or a chloride channel, which functionally results in the same thing, which is basically hyperpolarization. When potassium channels open, potassium leaves the cell. You're losing positive charge. The cell gets more negative. When chloride channels are open, chloride comes into the cell, bringing more negative charge. Same thing. You're causing an increase in negativity, or what we call hyperpolarization, and that is inhibitory, and that is called an inhibitory postsynaptic potential, an IPSP. And those again are just the rules as stated again: depolarization, excitatory, hyperpolarization, inhibitory. So then how do we take all these graded potentials and cause the postsynaptic cell to do something or tell it to not do something? And so this is the process of what we call summation. So the cell over here, the postsynaptic cell, is in essence listening to the input of all these presynaptic cells, some of them excitatory, some of them inhibitory. And it's going to take all of those responses and sum them up. And so basically, if the sum of all those responses creates a depolarization that reaches the threshold about 10 millivolts above the resting membrane potential, you will have a generation of an action potential. Anything else that's going to be less than that threshold level, there'll be no response. Okay, and so if you have more IPSPs than EPSPs, the cell is likely to do nothing. It's been inhibited from responding. But if you have more EPSPs due to the opening of sodium channels or calcium channels causing enough depolarization that that threshold level is met, you will then have the action potential generated, and basically that action potential will be generated again here at the axon hillock. So the kinds of summation, or the things to remember about summation is now when we're talking about graded potentials is they're not all or nothing events. And so that's really shown down here where you can see there's stimulus occurring, but it's not enough stimulus to actually get to the threshold. Okay, and that's what we mean by graded potentials. They're not all or nothing. The cell is listening, it's getting depolarized a little bit here, a little bit there, a little bit here, a little bit there. But in this case, it's not enough until finally there is enough EPSP, excitatory postsynaptic potentials, that you do reach this threshold limit. And then you begin the action potential, which itself is all or nothing. And then you basically, again, have the generation of the action potential at the axon hillock here. Okay, and so all incoming signals will be processed. And so that's basically what we said here. And so we have two types of summation, a spatial and a temporal. So a spatial is basically the cell listening, so to speak, receiving input from several presynaptic neurons at different parts of its own cellular structure, as you can see over here. And so basically, you've got an input here, an input here. Some of them are excitatory and some of them are inhibitory, and it's going to sum up all over these spaces. Do we have enough to actually generate an action potential? Temporal is a little different in that you can have EPSPs where a single one of them is not enough to reach the threshold, but if you have a couple of them in a fast enough time point and a high enough frequency, one can ride on the shoulders of the other one right there, and then it basically increases in its amplitude and can reach that threshold and then generate the action potential. Now, the postsynaptic receptors, in terms of the opening or closing of ion channels, we have two different receptor types. We have ionotropic receptors, and these are basically ion channels, and so the receptor itself is an ion channel. And so the obvious example of that is the one we just talked about, which are the acetylcholine receptors. Okay, and so basically these guys, when the acetylcholine binds to that receptor, it opens up the ion channel that it's attached to, and again, this ion channel is a sodium channel and causes depolarization. It is an excitatory postsynaptic potential. Metatropic receptors, though, they bind to a receptor, the molecules do, and the receptor is not a channel. But what it does is it initiates a cascade that leads to the opening of ion channels. Now, because of the way this connectivity is arranged in the architecture, they respond slower, but they last longer. Okay, and so again, here is the acetylcholine receptor, and this is acetylcholine receptors. Unfortunately for you guys in learning all this, there's two basic types. There's a nicotinic receptor and a muscarinic receptor. The nicotinic receptor is the one that we're talking about for muscles, and basically this is the one here, nicotinic, and the receptor is itself an ion channel. So when it binds, the sodium channel opens and we get depolarization. The muscarinic you see here is actually just a receptor bound to what's called a protein complex called the G protein complex. And then what you have is dissociation of separate parts of that, and they move through the plasma membrane and then bind to an ion channel. In this case, in the muscarinic, a potassium. So you can see here that if it's going to bind to a potassium channel and open it, you're going to have the opposite effect. Sodium channels open. In nicotinic, you will have depolarization, excitatory responses. Potassium channels open in muscarinic, you have hyperpolarization and inhibitory responses. So it's very important to know that and be able to dissociate between these two types of acetylcholine receptors. Nicotinic, because they're excitatory, these are the kinds of receptors we have on our muscle tissues at the neuromuscular junction or the motor end plate. So very quickly then, how do you stop the activity of a neurotransmitter? And this is an extremely important concept because this is something that gets manipulated on a pharmacological level in a way to treat a lot of different diseases and neurological conditions. And so basically you can imagine as long as that neurotransmitter is in the synaptic cleft, it's going to be binding to receptors and doing whatever it does. So under natural conditions, these neurotransmitters are being cleaned up as soon as they're being released. And so that's shown here. We have a presynaptic axon, and what it's doing then is it's releasing the acetylcholine you see here, and the acetylcholine will then diffuse across the synaptic cleft and bind the receptor. But almost just as soon as that's happening, there is also the presence then of acetylcholinesterase. And acetylcholinesterase is basically bound here to the cell membrane of the postsynaptic cell. And so the acetylcholine will bind to the receptor, and then it will find itself bound to the acetylcholinesterase receptor, where it will be destroyed and broken into its two component parts, acetate and choline. And there it can no longer function as a neurotransmitter. And so this is very important. So what it means then is the response happens, it occurs, it's very efficient, but then it's shut down immediately because you don't want these things to have a prolonged response because then the cell can no longer regulate it. And so in this case we use acetylcholinesterase to stop the postsynaptic response almost as soon as it's initiated, so you get a very brief and a very efficient response, but it doesn't last any longer than it needs to. And so as I was saying, this becomes a very, very critical leverage point in terms of treating nervous conditions because we can modulate using pharmacological agents the synaptic interactions. And so what we call an agonist is something that will mimic or potentiate the effect of a neurotransmitter. And an antagonist is the opposite. It will block the action of the neurotransmitter. So one example that's probably easy to appreciate is morphine is an agonist. And basically it is a mimic and potentiates the endorphin receptor, and therefore that helps block pain, and therefore we use morphine as a very powerful drug to help people through times of great pain. Okay, so basically we have several kinds of neurotransmitters, and this is an extremely brief list in terms of the type and their functions. But in the central nervous system we have acetylcholine, and it's very important also in the action on the skeletal system, or skeletal muscle system. And again, for skeletal muscles, it's always excitatory. We have glutamate, which is an excitatory amino acid. It's used in 80% of the synapses in the cerebral cortex, so the very large sort of frontal part of our brain, where a lot of our powers of reasoning are apparently located. a lot of that is glutamate dependent. And we have glycine and GABA, which are inhibitory amino acids, and we also have a broad group called monoamines, and there we find amino acids such as serotonin, which is very important for mood, behavior, appetite, and also cerebral circulation, and then dopamine, which is extremely important for reward-motivated behavior and therefore implicated in a lot of pathologies involving drug addiction. And so this is also just to say how important some of these things are when we start affecting them with drugs or different diseases. And so strychnine, which you guys may know as rat poisoning, well, it kills rats because what it does is it blocks the glycine receptors required to get the diaphragm to relax. And so if our diaphragm is working to allow us to breathe in, and basically you have it sort of paralyzed so that you cannot relax the diaphragm, you've effectively shut down your ability to ventilate your lungs, and therefore you'll die of asphyxiation. So the problem with strychnine or rat poison is that it doesn't just kill rats, it also kills anything that eats the rats. And in fact, you may have heard recently there's a very sick mountain lion running around the hills of Hollywood by the Hollywood sign because it probably has indirectly ingested rat poison. And the loss of GABA neurons in the cerebellum has been known to be associated with a very debilitating disease known as Huntington's disease. Serotonin is very important because drugs you may have heard of, pharmacological agents such as Prozac and Zoloft, they make people feel better because they shut down the reuptake process. And so we're talking about with acetylcholinesterase that breaks down the neurotransmitter. The other way presynaptic cells can control that is not necessarily breaking them down, but once they release the neurotransmitter, they then take it back up again and remove it from the synaptic cleft. That's the case with serotonin, and if you inhibit that process of reuptake, you allow the serotonin to have a much longer and more dramatic effect on those postsynaptic neurons, which generally allows certain people to be happier. And that's how these kind of drugs work. And then dopamine is also extremely important for motor control. And in fact, it's been linked to very important diseases in terms of the quantity of dopamine. Basically, having too little dopamine is associated with the neurodegenerative disease, Parkinson's disease, where you get a lot of shaking and loss of muscle control. And then if you have too much, that's been correlated with cases of schizophrenia. So you can see overall the importance of the regulation of these neurotransmitters in terms of being able to regulate their effect on the postsynaptic neuron and how long they're in the synaptic cleft. And by tweaking that, you can have very positive outcomes in terms of mood and behavior, or you can have very negative outcomes too, as you can see in the cases of things like Parkinson's and schizophrenia and the use of rat poisoning. Okay, so that's it for that. I just wanted to again go over those points in a little more detail since we were rushed at the end of class on Wednesday. So I hope you guys look at this and get a better understanding, and as always, any questions, come by and see me during office hours and we'll talk it out. All right, I'll see you guys on Monday.